Hello, this is Amanda Welch welcoming you to this Bite-Size Bio web seminar, which today is sponsored by Bio-Techne. Today's presentation is titled Multicolor Fluorescence Immunohistochemistry Using Primary Antibodies Raised in the Same Host Species, and is being presented by Dr. Alexander Kaluzhny from Bio-Techne. Alex graduated in 1981 from the Biological Department of the Saratov State University in Russia. After graduation, he performed research on the effects of narcotic drugs on the morphogenesis of early embryos, which he defended as his PhD thesis in 1987. In 1992, Alex was invited to join the Department of Pharmacology at the University of Minnesota, and in 1998, he joined R&D Systems and helped with setting up the IHC ELISA Spot lab, which he is managing now. He assisted with teaching a histology course at the University of Minnesota and is regularly attends scientific meetings, giving IHC and ELISA Spot presentations and workshops. He's the author of 31 papers in peer-reviewed journals and 16 book chapters, and has served as the editor of 3 books with Springer Humana Press. As always, we will have a question and answer session after the presentation, so please type any questions that you have into the questions box, which appears on the right-hand side of your screen, and I'll put them to Alex at the end. So now, over to you, Alex, for the presentation.
Hello, thank you, Amanda, for such a nice introduction. Today the topic of our presentation is several techniques we will go over, how to use multicolor fluorescence immunohistochemistry when we're using primary antibodies raised in the same host species. And quite often, this is quite challenging to do this technique or actually do immunohistochemistry, because quite often we're limited to just one type of host species. In academic labs, rabbits are very popular hosts to make antibodies, and lately there are facilities making mouse monoclonal antibodies. And when we want to do multicolor fluorescence, we're stuck. There are serious limitations how to use these antibodies from the same host species to get nice staining when we don't have any cross-reactivity of secondary antibodies with our primary antibodies. Today I will be covering 4 different techniques. Some of them you probably may know about, and if there's something that you don't know about, we'll be happy to share our methods with you and hope they will help you in your research. So before we go into the depth of this discussion, we will need to refresh our memory and go with terminology. So IHC stands immunohistochemistry, and it's the term when we want to emphasize that we're staying tissue sections. Whereas immunocytochemistry, or ICC, we use this term when we're trying to emphasize that we're staying in cells as cytosmeres or cells in culture which are fixed with fixatives. And quite often this term is used interchangeably, but we will actually delineate these 2 terms, because that's very important, because not all the technique for multicolor fluorescence can be used on both, on tissue sections and cells. And then another interesting word that's as a buzzword, we can call it, it's immunofluorescence, and this is pretty much defines the detection method rather than the sample type. So immunofluorescence can be done on tissue sections or on cells, and that's why it's applicable to both types of material. So this is a typical workflow of the IHC protocol. First, we have to fix the cells or tissues, and fixation done either using formaldehyde or as a cross-linking fixative, or precipitating fixatives like ethanol, acetone, or some other chemicals. And after fixation, cells and tissues are, of course, washed in buffer or any other solutions, and then they are ready to be incubated with primary antibodies, monoclonal or polyclonal. After incubation with primary antibodies, we are incubating tissue samples or cells with the secondary antibodies conjugated to fluorescent dyes. And then after it's done, we examine tissues under the microscope. So that's a conventional workflow of a typical IHC experiment. Now, there are several factors which will determine the success of IHC experiment. And first of all, we have to preserve tissue antigens. They have to be available to antibodies that we will use to detect them. We, of course, have to preserve cell and tissue morphology, because if it becomes amorphous, it will be hard for us to identify the localizations of the antigens. Of course, the key another key parameter is the quality of primary antibodies, because no matter how good our morphology preserved and how good the antigens preserved, if we are using, or if we are limited to using antibodies of low quality, then our experiment may not work out. And quality of antibodies, I mean it's their affinity and their specificity. And then, of course, another important factor is the quality of secondary antibodies and other detection reagents, because if secondary antibodies are prone to non-specific interactions with tissues, in addition to specific signal, we will see non-specific one, and it may obscure the specific staining of the antigens. Why fluorescence? Fluorescence is a perfect technique because we can look at multiple antigens in the same tissue. And we're talking about the antigens that may overlap or non-overlap. For example, you of course know about technique when we use enzymatic detection using HRP and diaminobenzidine, and that's chromogenic detection. But unfortunately, if we combine two chromogenic techniques using, for example, HRP substrates and alkaline phosphatase substrates, these targets ideally need to be localized to different compartments of the cell. So we call them non-overlapping, because if they overlap, it will be hard for us to analyze the expression of each antigen in particular, because they may mask each other. In fluorescence, we don't have this, because we are looking at individual channels, and then we can digitally overlay and merge them and see all the colors at once, or just see as many colors as we want. Fluorescence allows to get very high resolution and contrast compared to chromogenic detection. And like I said before, it's a perfect example of using perfect technique to use overlapping, to detect both overlapping and non-overlapping antigens. It allows for quantitative image analysis, so we can measure the fluorescent signal, and that may be translated into the amount of the antigens expressed in cells and tissues. And what's also more important is that using fluorescence, we can do 3D reconstruction of the tissue section or antigen distribution in the thick tissue. And that's quite often is done by using, for example, whole mount incubations, or another way of calling this technique or using it is called free floating sections, when unlike 5 or 10 micron thick sections, we can use very thick 50 to 250 micron thick sections, and then do Z-stack using confocal microscope, and then reconstruct them and get a 3D object. This is to refresh our memory about the fluorescence detection. For example, here we talk about 3-color fluorescence, and this is the conventional technique when we have ability to use antibodies, primary antibodies raised in different host species. And here we have rabbit primary antibody, mouse, and goat. And then we are detecting them using secondary antibodies raised against rabbit, against mouse, and against goat. So there's no expected cross interference or cross reaction of, for example, anti-goat secondary antibody with the mouse primary antibody, or anti-mouse antibody with goat. So we're doing good. So this is a very convenient and easy way to do multicolor fluorescence. But now let's say if what is going to happen if we have the primary antibodies raised in the same host species and we want to detect them using different colors. If we use, for example, primary rabbit antibodies and then anti-rabbit secondary antibodies conjugated to different fluorophores, all the secondary antibodies will cross-react with primary antibody. In the end, we will see complete overlap in colors, and we will not be able to tell which color depicts which antigen. That's the major problem. That's quite easy to understand. There is a technique that's been actually described a long time ago, and has been published in the Journal of Histochemistry and Cytochemistry, when people can use tyramide signal amplification technique and so-called antibody stripping protocol. The way how the whole reaction works, before we go into the protocol itself, I would like to remind you what tyramide signal amplification allows to accomplish. So we incubate tissue sectional cells with primary antibodies, followed with anti-primary antibodies conjugated to enzyme, host radius peroxidase, HRP. We'll get this sandwich here. And then we add tyramide, labeled with fluorescent dyes. HRP will activate this tyramide, and it can now bind to tyrosine in proteins to its phenol group, and it binds so tightly that at the end we actually don't need the presence of primary and secondary antibodies. And the activated tyramide binds to these phenol groups and proteins, not only in the protein of interest, but in proteins adjacent to the protein of interest. That's how tyramide amplification works, so to speak, in a nutshell. Now, let's look at the protocol when we are using TSA chemistry and we do so-called antibody stripping. We do incubation with the first antibody, followed by incubation with secondary antibodies conjugated to HRP, and addition of tyramide, let's say labeled with red fluorescent dye, and we will get a staining here. So now we have tyramide bound to the tissue, which depicts the localization of the antigen. So what we do next? Next, we are removing primary and secondary antibody. We're doing what's called stripping them away. This can be done using either low pH or high pH buffers, or using the microwave to detach the antibodies and wash them off. So now what we get in the end, we got the antigen and only these dyes attached to the tissue. What we do next? We repeat the same cycle here. We incubate now with another primary antibody, again, from the same species, like rabbit, okay, and repeat the whole reaction again. But now our tyramide is conjugated to fluorescent dyes of different color, okay, and we do the same. Then we remove, strip off primary and secondary antibody, leaving only the bound tyramide conjugated to fluorescent dye. And we can repeat it another round and with a different dye. And in the end, we will get several different colors. And again, we use here antibodies raised in the same host species. That's briefly how this reaction works. There are pros and cons in using the TSA antibody stripping protocol. Pros. TSA is known to be incredibly sensitive technique, which means that the concentration of primary antibodies may be lower than what we usually use in our conventional immunohistochemical protocol. When we strip the antibodies from tissue sections, we eliminate the risk of cross-reactivity between primary and secondary antibodies that we will use in subsequent cycles. Here we can, if we stretch our imagination, we can pretty much use virtually unlimited number of multiplex tissue targets. There are serious cons to this protocol. The protocol is long and cumbersome. It's tedious, and someone needs to be not a novice, but know and experienced in doing IHC. Subjecting tissue sections to stripping treatments may actually affect the integrity of tissues and their morphology, which in the end may actually lead to having tissue sections which are absolutely unusable. And this protocol is more practical for IHC to stain tissue sections rather than for ICC using cells, because when we strip these antibodies, cells are more vulnerable to destruction, and we pretty much can lose the material from our slides and then end up with nothing there. So besides TSA Another protocol to do multicolor fluorescence is the, we call it non-TSA based protocols, and they are direct and indirect multicolor fluorescence. Direct detection is the detection when we can modify primary antibodies by conjugating them to fluorophores directly. This is very convenient, very fast, very easy to do protocol. And then we do indirect detection when we modify primary antibodies by conjugating them to small hapten molecules, and then use anti-hapten secondary antibodies conjugated to fluorophores. So we want by all means to avoid using secondary antibodies that can interact with the host species with our primary antibodies. That's why we use these small haptens attached to them. Let's look at these protocols and go over their pros and cons as well. This is a direct detection in IHC, direct workflow. So, like I said before, antibodies conjugated directly to fluorescent dyes, and then we can pretty much mix them and have all three antibodies in one cocktail and incubate tissues or cells with these antibodies, and it's a very short protocol. We get detection easily and get results pretty fast. What are the pros? It's a very short protocol. If you stain cells, for example, and it requires only incubation for 30 minutes, in 30 minutes you have all your samples stained. If for tissue sections, you can do it also for maybe a couple hours room temperature, or do it overnight at 4 degrees C. And here, if you can combine and conjugate primary antibodies to different dyes and have means to analyze these dyes or visualize under the microscope, so you have all necessary lasers on the confocal microscope, we have all the cubes on the conventional microscope, you can easily see them. So you can pretty much have a large number of targets detected. And it can be used easily for both IHC and ICC because there are no harsh treatment conditions for cells. Cells will remain intact. There are cons to this protocol. So it works good for abundant tissue targets, because when you don't have a large number of fluorescent dyes detecting the antigens of interest. And that's why the target should be abundantly expressed to be visualized. A conjugation of fluorophores to primary antibodies may impair their affinity and result in weak staining. Like any conjugation of primary antibodies to any hapten or fluorophore, it may interfere with binding sites on the antibody. And of course, it requires resources to perform conjugation. So you have to either find a company that can do this service for you, or buy conjugation kits and do it by yourself, and then analyze the yield of conjugated antibodies. So these are the obstacles that should be kept in mind. I mentioned that the direct detection may not be as sensitive as indirect detection, and there is chemistry behind this. How many dyes can be conjugated to the primary antibody? Usually it's about between 4 and 8 molecules. If it's more, then these dyes may quench each other, and instead of having bright fluorescence, fluorescence will become dim. So the signal will be weak and may be undetectable. When we, for example, and then when we compare it to indirect detection, we have to keep in mind and remember how many secondary antibodies can bind to primary antibodies. And usually we talk about that between 2 and 4 molecules of secondary antibody per 1 molecule of primary antibody. So let's say if we have 4 dyes conjugated here, and we have 4 dyes conjugated to secondary antibodies. In this case, if we have 3 secondary antibodies bound to primary antibodies, we have 3 times more fluorophore. Signal will be 3 times brighter than what we can accomplish doing just direct conjugation. That's what has to be kept in mind. Now we will talk about indirect multicolor IHC using primary antibodies conjugated to haptens, and using antibodies, secondary antibodies, anti-hapten, hapten-specific secondary antibodies. So they will interact with haptens rather than with antibodies per se. let's take a look at this protocol, and I will walk you through how this can be accomplished. We can conjugate primary antibodies to haptens, which you have heard about, so-called biotin. Okay, it's known, used a lot of quite often in immunohistochemistry, or we can use the deoxygenin to put on the primary antibodies. Deoxygenin is not the animal protein, it's found in plants. That's why if we use anti-deoxygenin secondary antibodies, there's virtually no cross-reactivity of antibodies or no background staining with cells and tissues. So now we have two primary antibodies, one conjugated to biotin and another one to deoxygenin. How about the third one? This one will leave unmodified. This one is going to resemble the conventional IHC protocol when we use primary antibody plus secondary antibody. Of course, all these secondary antibodies conjugated to different fluorescent dyes. So how this protocol works? First, we incubate tissue section with this primary antibody, followed by incubation with anti-primary secondary antibodies conjugated to this fluorophore. This is the conventional protocol. After all washes, we now can incubate tissue sections or cells with primary antibodies conjugated to biotin and to DIG, to antibodies, followed by incubation with anti-streptavidin conjugated to fluorescent dye and anti-DIG secondary antibodies conjugated to this fluorophore. We can mix them together and incubate. They are not supposed, and they will not cross-react with this primary antibody because they are raised against different molecules. That's how we got this multicolor staining using antibodies raised in the same host species. This protocol works nicely with different primary antibodies raised in different species. For example, here we talk about, it's an example of using mouse primary antibodies, and that's staining in rat brain. Here we incubate first with calbindin D primary antibodies, and they are unmodified. And then we do incubation with primary antibodies conjugated to biotin, anti-synaptotagmin, and CART primary antibodies conjugated to DIG, followed by using anti-DIG secondary antibodies and streptavidin conjugated to another fluorescent dye, and of course followed by counterstaining using DAPI. Now we have all three colors here, specific colors. Color number 4 is DAPI, and then we can merge these colors and get this multicolor picture shown here. So this technique works not only with mouse primary antibodies. We tested it, and it works nice with goat primary antibodies. This is, here we're showing staining of a rat brain cerebellum. You can see this large neurons, the Purkinje cells, Purkinje neurons. And now again, we first incubate tissue sections with unmodified primary antibodies conjugated to E-cadherin. Primary antibody, unconjugated primary antibodies against the E-cadherin, followed by anti-goat secondary antibodies conjugated to this fluorophore. And then we incubate tissue sections with the mixture of primary antibodies. It's neprilysin conjugated to biotin and netrin conjugated to DIG, followed by incubation with anti-DIG and streptavidin conjugated to the fluorophore. And then we can get this picture. Of course, there's DAPI, always used for counterstaining. Now you can see that all the colors, what we see individually on these images, you can actually recognize them and see them here as well. And the third example I'm going to show you is using rabbit primary antibodies. And this is very important because quite often we know that for known reasons, antibodies against phosphorylated targets, phosphospecific antibodies, somehow are the best when they are made in rabbits. And that's a good example of using such antibodies and doing again this conjugation of primary antibodies to to biotin and to DIG and combine it with unlabeled primary antibodies. Again, all the colors are nicely seen here. And when we merge this image, merge all the channels, we got this multicolor staining here. This is the red dorsal root ganglia. That's where we show the staining. Pros of this technique: there's no cross-reactivity of secondary antibodies with irrelevant primary antibodies, because our second round of incubations, we do not add secondary antibodies against primary antibodies. They are against these small haptens. It's like biotin and also deoxygenin. Conjugation to biotin and deoxygenin can be done by the end user. There are kits sold by different companies that allow the end user to conjugate these antibodies. Protocol is very easy, simple, and easy to run. So anyone can do it as long as they follow the sequence of incubations. And this can be also used for both IHC and ICC, because there's no any post-processing of tissues or like stripping antibodies or any harsh treatments. Cons to this protocol: it's limited to 3 tissue targets. If you want to do more than that, it should be, it can be expanded to 4 targets, given that one of the targets is highly abundant, and you can use pretty much directly labeled primary antibody. It may require quenching tissue endogenous biotin. You know that we're using here streptavidin that's supposed to bind to biotin on the primary antibody, but some tissues may have endogenous biotin. That's why it's required to quench it. Otherwise, streptavidin conjugated to fluorescent dye may cross-react and get non-specific staining. And of course, it requires the resources to do conjugation. Resources need have money to buy these conjugation kits, or if you want to do it by yourself, or find a company that will do it for you. Now we will talk about the modification of this protocol. I have talked about the anti-hapten detection when all primary antibodies conjugated to haptens. And these haptens are different from biotin and deoxygenin. They are proprietary, and this technique has been recently developed by our collaborator company CellIDX, and this is pretty much what they do. They came up with the proprietary haptens. We don't know their structure, and we don't know how they do conjugation to primary antibodies. And they conjugate primary antibodies with haptens, and then you do staining incubation with secondary antibodies raised against this particular hapten. Currently, they have in their portfolio 4 different haptens and 4 different anti-hapten secondary antibodies conjugated to different fluorescent dyes. And the beauty of this technique is that when all primary antibodies conjugate to different haptens may be mixed together in a solution, a cocktail, and you can incubate your cells and tissues with this cocktail, followed by incubation with 4 secondary antibodies which are specific for each hapten. And this is the example of this technology. Here is the staining for progesterone receptor, estrogen receptor, HER2, and Ki67. And you can see that all these channels can be merged together, and we can see the multicolor tissue staining. Interesting enough that actually if you can actually combine staining on 2 adjacent sections and get like 7 or 8 different targets, detect them simultaneously. And that's an example. When 2 adjacent sections were stained with the first set of primary antibodies conjugated to haptens, and another slide is stained with next set of primary antibodies conjugated to fluorescent dyes. And then images can be collected pretty much as black and white and then pseudo-colored. But then both staining from slide 1 and 2 can be merged together, and that's given this, well, somehow messy image, but if we talk about multiple colors, they are all here. So this technique is also also available to researchers to do multicolor fluorescence. Pros to this technique: Okay, primary antibodies are species independent, and it's a standard two-step staining protocol, like what you would do in conventional IHC. There's no non-specific staining, okay, because we don't use secondary antibodies against the species of the primary antibodies. Simultaneous detection of 4 biomarkers and plus dipase counterstain on the same tissue section. And actually more haptens are emergent, or they make more different haptens, then pretty much this technique has unlimited potential for multiplexing. And it can be used for both IHC and ICC. The drawbacks or cons to this procedure is that this is a proprietary technology, and like with any proprietary technology, the conjugation to haptens can be done only at the site of cell IDX who know how to do this conjugation, who have IP on their conjugation chemistry, as well as they will provide the anti-hapten fluorescent secondary antibodies. So no other source of the secondary antibodies is available to the researcher. So that was technique number 3, and now we are into technique number 4, and this is an interesting chemistry because it combines both TSA technique, what we talked about before, and non-TSA technique, and that's going to be like a sequential procedure. Here's how it works. We know that TSA technique is incredibly sensitive. It means that, for example, we can use 200 less concentrated primary antibody and detect with TSA. If we use in a conventional IHC 200 times diluted primary antibody from the, its optimal concentration, our secondary antibody will not recognize it. They will not see it. Sensitivity will not be adequate and will pretty much get no staining. And knowing this phenomenon, we can now combine both TSA and non-TSA technique. Here's how it works. First, we incubate tissue section with the primary antibody diluted so much that it cannot be detected with the secondary antibody in a conventional IHC. Okay, and then we do TSA technique here. Okay, we got staining, activation of tyramide fluorescent conjugate by HRP. Tyramide will bind to phenol groups in tyrosine and proteins, and we'll get our signal. Second phase, we are going to incubate the same tissue section with the primary antibody, also rabbit, from the same host species, but in a normal concentration, normal optimal working dilution, which can be picked up and detected using the secondary antibody, like here. There probably cross-reaction of the secondary antibody here with these primary antibodies, but because the concentration of primary antibody is so low, we will not be seeing the signal or binding of this secondary antibody to primary antibody will not generate any visual signal. It will only generate the signal applicable to this primary antibody using a high concentration. That's how both techniques can be combined together. And believe it or not, this technique is not new. It's been reported many, many years ago. And actually, this is I will show you the screenshot of the paper published in 1996. So the optimal dilution here is, again, go over this technique. Optimal dilution of primary antibodies for detection by TSA is 100 to 200-fold more dilute than in standard IHC protocols. All these high dilutions, okay, at these high dilutions, most primary antibodies are undetectable by conventional fluorophore conjugated secondary antibodies. And two-color is performed by using first TSA to detect first highly diluted primary antibody and fluorophore conjugated secondary antibodies to detect second primary antibody at their normal dilution. Pros, this technique, it's a very simple protocol. No need to strip primary antibodies away like we've done with TSA technique talked about earlier, and can be used for both IHC and ICC, so because there are no treatments to cells and tissues. Cons to the procedure, limited to 2 antigen detections. Only 2 antigens can be detected, you've seen it here, and requires a preliminary testing of optimal dilution of primary antibodies to make sure in TSA phase we are using the primary antibodies that will not be detected by second cycle secondary antibodies. So now that's how all these 4 techniques work, and that's a paper I mentioned to you. It's been published in 1996, and a presentation has been made at the meeting of the Histochemical Society meeting, and can be easily used to do this 2-color staining. So let me summarize what we just talked about. So multicolor IHC with antibodies from the same host species is easy to do. Multicolor staining can be done with direct or indirect techniques, either alone or combined together. Primary antibodies have to be tested first in single IHC staining with all positive and negative controls. And the specificity and affinity of primary antibodies is of critical importance. So we have to remember, no matter how good and sensitive our detection techniques are, we need to make sure that the primary antibodies are of high affinity and specificity. Okay, let me stop here, and I'll be happy to answer your questions. Thank you.
Thanks, Alex. That was an excellent presentation. We have a few questions from the audience, and if anyone else has a question, please feel free to post it in the questions box that appears on the right of your screen. So our first question is from, I apologize if I mispronounced your name, Maria, and they want to do multicolor immunohistochemistry in mouse lung tissue, FFPE, but he's getting extreme auto from tissue. So do you have any suggestions about options to remove autofluorescence?
This is a very good question, and actually I've been thinking about giving a separate webinar on how to get rid of endogenous autofluorescence, because this is a very serious issue. Very good question. Yes, you're correct. When you have endogenous autofluorescence due to fixation or overfixation or due to the presence of substances in the tissue, it's incredibly difficult to do immunofluorescence, because this endogenous fluorescence will obscure specific signals. There are several treatment techniques how to quench endogenous autofluorescence. None of them is going to quench autofluorescence completely, but it helps. And it depends on the tissue you use. It may be working better in one technique than in one tissue compared to the other. And please take a look, do Google search. People use, there's a paper published using Sudan black B dye to stain tissues before you're doing staining with the fluorescent detection reagents. Sudan black B. It binds to, actually Sudan black B is used as a conventional common histological dye to stain lipids in tissue. And somehow when it stains, it blocks endogenous fluorescence. And people use it, and it may work in your case. So easy to do. I don't have a recipe in front of me, but you can easily find it, like I said, if you Google and you go PubMed and do this search. Another technique people use also, you may Google it and find people incubate tissue sections with copper sulfate. Freshly made copper sulfate, I believe in citrate buffer. Also do Google search and you will find a recipe how to do this. So these are the 2 techniques I can think of. We are developing, working on the solution to quench endogenous fluorescence, and the project is underway, but we have mixed success, but we're still working, and I hope that someday we'll come up with a solution how to quench it and allow people to use multicolor fluorescence, for example, in human tissues which are known to be autofluorescent.
Great. Yes, I hope so. And Amir has a question asking about, in a double or triple stain with the same host primaries, what is a robust negative or positive control, or both?
Oh, good question. Robust, negative, and positive control. Negative control is you don't expect to see any staining, and actually let me see how you can do this. Well, you negative control pretty much if you do multicolor. You need to make sure that your secondary antibodies is not binding to your first primary antibody. So by other words, your secondary antibody is not binding to irrelevant target. And the way to do it would be, for example, let's say we're talking about DIG-labeled primary antibodies. First, we incubate the tissue with the unmodified primary antibody, and then instead of going with the with its secondary antibodies, we incubate the tissue with the anti-DIG labeled secondary antibodies. And in this case, you're not expecting to see any signal, any staining. That's going to be a good negative control. Positive control is easier to do because you are doing first, you do single color experiments, and you compare your Hapten conjugated primary antibody. For example, we'll take the DIG labeled primary antibodies again. You compare your staining, your signal, with when you do single color with DIG conjugated primary antibody and with unconjugated primary antibodies. Your signal's supposed to be the same from the standpoint of its intensity and its localization. That's how I see doing these controls.
Okay. And now Sally has a question asking if you could suggest within how much time post-addition of the secondary antibody should the fluorescence be determined.
So the incubation time, this is my understanding. How long to incubate with secondary antibodies to get a signal?
Right.
Usually incubation is done between 30 minutes and 1 hour. Some people do 2 hours, but you have to watch, and again, you need to run the control to see that there's no non-specific sorption of secondary antibodies the longer the incubation is going on for. Okay, so you may do on cells, usually we do 30-minute incubation, but cells is a different story because cells are dispersed and there's no need to give more time for better penetration of the secondary antibodies into the tissue, like when we do immunohistochemistry. So this is faster. With tissue sections, it can be, again, depends on the antigen. If it's abundant antigen, then you can take 30 minutes or 1 hour, but if not, you go for 2 hours. But again, I assume you have a control when there's no primary antibodies and you add just secondary antibodies and compare them side by side to make sure there's no non-specific sorption of secondary antibodies which imitates or mimics somehow the specific signal.
Okay. And Maria asks again, wants to know why is it so hard to find DIG conjugated or Bio-Techne conjugated antibodies for immunohistochemistry?
Well, it's hard to find for reasons not related to big science. When companies are making antibodies, right, and they are, let's say they want to do conjugation of every antibody they made to biotin. It's not just the conjugation, that's the quality control that needs to be done on these antibodies, there's storage conditions, and pretty much it's a lot of work. That's why just to cut costs of production, companies are not doing it often and may offer some antibodies conjugated to biotin, but that's a costly business which many companies don't want to go into. It's easier to get, for example, if you're interested in using biotin conjugated primary antibodies, it's easier for you to contact the company and say, okay, I need this primary antibodies conjugated, please conjugate them for me, and they may charge you additional. They will do it on their own and get you such antibodies. So it's a commercial rather than scientific reason. That's how I would say.
Okay. And Amir asks, would you dismiss any sequential staining protocol with primary antibodies of the same host? So that is first primary, first secondary, wash, second primary, second secondary, and so on.
Unfortunately, there's a high risk of cross-reactivity of irrelevant secondary antibodies to irrelevant primary antibodies. Let's put it this way: no matter how good the washes are, you will still get you may get association interaction of secondary antibodies with irrelevant primary antibodies. That we checked it many times, and it may sound illogical that, yeah, if we do all the washes, we should get rid of all these non-specific sources of signal, but somehow it's not working, and the secondary antibodies will find their way to irrelevant primary antibodies. So it's not a good way of doing it.
Okay. And then, I'm going to, Zach, I'm sorry, I cannot pronounce your name, says that they've had a lot of trouble with TSA and never got it to work in their in situ stainings. So what do you think might be the reason for this? And do you have any advice for basic troubleshooting? And just for extra information, it's a non-model insect organism whole mount.
Well, first of all, I don't know what troubles people have with TSA, and I would like to answer this question if you just can send me this, the email, or somehow contact me and give me the details of what you're doing, details of your protocol, so I can do in a more specific way and give you suggestions.
Okay, so if you want to pass, I'm sorry to butcher your name, but Tzak, if you want to pass along your email to me, you can type in the questions box. It just goes to me, and I can pass it along to Alex. So now we have one, I think you might have mentioned this, but Prashad asks if there's any publications using multicolor immunohistochemistry fluorescence using the same species primary antibody. Oh, so maybe not. So he's asking if there's any publications using multicolor immunohistochemistry fluorescence using the same species primary antibodies in FFPE sections on cancer specimens.
Well, what I have shown in my presentation, well, I have not seen the publications because that's kind of novel, but you have seen that slide when that company came up with the proprietary Haptens, and that's what this guy is exactly doing. They're doing multicolor for pathology diagnostics, and unfortunately, it's so new. It's just the whole technique is in its infancy, and there are no papers available, but you can contact this company and ask them directly, so how they do it and if they want to do it for you. Of course, given that you will use our antibodies made by R&D Systems, so they will conjugate them for you, and you can use anti-Hapten secondary antibodies.
Okay, and now Amir asks if you have any suggestions for antigen retrieval, that is like 1% SDS for 5 or 10 minutes or something like that.
Antigen retrieval is not actually, I would call it voodoo science, because no one knows how it works, and we use different antigen retrieval protocols. We use TRIS buffer pH 9, we use citrate buffer pH 6, and I don't think I can give you recommendations. It's pretty much whatever works the best when you get the strongest signal with the lowest background. We get very good success with TRIS pH 9. That works pretty good, and we use it quite often in our experiments here. So.
Okay, and then Amir also asks if you can touch on frozen sections.
I guess we're talking about frozen sections which are fixed with formaldehyde. So yes, when we talk about frozen sections, and I don't know, we talk about retrieval, we know that antigen retrieval on frozen sections versus paraffin-embedded tissue sections is not working because frozen sections are very sensitive to retrieval. They may get damaged by retrieval treatment. Speaking of staining, there's no difference in staining frozen tissue sections versus the paraffin-embedded tissue sections. We know that actually the papers published that in frozen tissue sections, antigens are preserved better than in paraffin-embedded ones, and that's understandable because when tissues are embedded into paraffin, the conditions are pretty harsh. It's high heat and all the solvents used to do it to embed tissue into paraffin. Whereas in frozen tissue sections, you fix tissues and then cut them on the cryostat and do staining. They are more gentle, I would say, And of course, when you decide, let's say, do TSA stripping protocol, it may damage tissue pretty bad, and after the first round, you will not see tissue on the slide anymore. That's why this protocol, TSA stripping, is recommended for paraffin-embedded tissue sections rather than frozen. So they're more sensitive, but if you do incubation when there's no treatment required, like even with, I have shown you tissue sections in the red brain, they're all frozen tissue sections, and they can be used nicely with this protocol. We have like a sequence of conventional with the DIG and biotin labeled primary antibodies.
Okay.
I hope I answered the question.
Okay, and so it looks like you have. Now, Inez asks, on the topic of quantification of proteins with immunohistochemistry, any advice on how to calculate the amount of sections we will have to count to make sure that our results are representative of the entire sample?
Well, the answer is the more the better, but of course it's not possible practically. And usually what we have done to do for quantification, we're looking at around 10 tissue sections, and then averaging the signals, and then getting the results into the table. That's what we do. So one section is not enough. Two sections, better than one, but still may not be quite good. And three, some people say the magic number of three, not less than 3 sections. Well, probably, but we prefer to go with a high number of sections, so we feel more confident that our quantification is accurate enough.
Okay, that looks like, let me check and see if there's any more questions. So that looks like the end of our questions. Now, oh, sorry, so there's one more. Are there any drawbacks in mixing all hapten conjugated primary antibodies together for adding to tissue sections?
We have not seen any obstacles, any problems when we mix all these hapten conjugated primary antibodies. So I would say no. We don't see any problems doing this.
Okay, and then Amir asks again, how would you quench transgenic fluorescence signals to be able to restain on in the same channels on a frozen section?
How well, it depends what technique you're using. If you're using TSA technique, you cannot quench the signal. You cannot quench it. You can only quench or remove that staining when you do conventional or you use antibodies conjugated to fluorescent dyes, and then you can strip them away using the low pH buffer. Same what you do when you do purification of antibodies on the column. So that's how it can be done. And this technique is not new. It's been done. I've seen a paper published in 1974 when people are doing it and stripping away this antibodies and then restaining tissue with new set of antibodies. So you need to strip away using low pH buffer.
Okay. Well, that looks like the last of our questions. So that brings us to the end of the seminar. So thank you again, Alex, for a very illuminating presentation and a great discussion. And thanks also to our sponsor, Bio-Techne. And finally, thanks to you, the audience, for taking the time to attend and listen in. If you've enjoyed the seminar and would like to view the video recording of the session, please visit the seminars page on bitesizebio.com. It should be available within the next 24 hours. There you can see the other webinars we have lined up for you in Bitesize Bio's Webinar Festival. So until next time, good luck in your research and goodbye from all of us at Bio-Techne and Bitesize Bio.